So we're at the end. Today is the last, sort of didactic section of the class. On Thursday, I will be sending out your final exams. You will send me a sheet of paper that is your study guide for the exam, and I will send you the exam back on Thursday. I will expect you to turn in the exam when you are finished with the exam. I'm going to ask you not to cheat. I'm going to ask you not to consult your friends. I'm going to ask you not to consult previous years, and do your best to try to take the exam in a few hours and don't kill yourself over it. But I'm not going to require you take it Thursday or Friday or Saturday. You take it when it is appropriate for you to take it. If at some point or another, when you are finished with the exam, I would like you to send it to me. And if you have any outstanding homeworks, you should send me those homeworks too when you send me the exam. When you send me your final exam, I'm going to assume you are done with this class, and you're going to get whatever grade you've gotten on the exams and with whatever homework you've done up to that point. So don't send me your exam until you intend me to assign you a grade, and I will assign you a grade sometime after you send the exam in. And if for whatever reason, because you've been busy with qualifying exams and you're continuing to be busy with qualifying exams and you're just not quite caught up, if you do not send me your exam back, I will not assign you a grade. I will leave it as an incomplete, and you in the administration and me, and we'll figure it out some other time. We'll let you finish the class. We'll figure it out. We'll all be adults here sooner or later. If you don't want to go through the process of having an incomplete on your record for some period of time, try to send me back the exam sooner rather than later, because I'm sure the administration wants to close out grades in a week or two. And God forbid, if you are graduating or something this term, definitely try to send me your exam soon so that we don't interfere with any of that crap. But most of you in here are graduate students who are not about to leave the university. So I don't think we'll have any of those type of concerns. But again, just act like an adult. Do your best. Don't kill yourself. Try. And if you guys do anywhere near like you did on the midterm, expect the worst thing that's going to happen to you in this class is probably a B+, and the absolute worst thing is going to be a B, because none of you were even near B work really on the midterm, and the vast majority of you were doing somewhere between a solid A and embarrassing me at how well you were doing compared to when I thought my exam was hard. You didn't, so we will. We'll see about that. The exam will technically cover this class, so we'll have probably a question or two about some of these readings. It will potentially cover things from the first half, but not really. Bring your cheat sheet from the first half just for your own benefit, but it will only cover things from the first half to the same extent that, we have never, ever given up the definition of a covariance or an expectation, or, all of those have been important all the way through. Hardy-Weinberg's been important all the way through. And if you're struggling with any of that stuff, God forbid, the quantitative genetics is going to be miserable. So, but keep all that, keep your old sheets, but assume all the questions are really coming from the last 4 homeworks and the 2 papers we are going over today. Are there any other questions about class procedures, timings, anything like that? The basic answer is you be responsible and nothing bad's ever going to happen. That's just it. We're here to learn. Oh yeah, you probably should have gotten a class evaluation form. If you have 2 or 3 minutes, fill it out and then send it to Levi so that I can't see who it is, So when you say you hated me, I won't know which one of you it is that hates me, and probably Levi won't even say that, won't even send them to me until after the class is over. So there'll be no danger of me retaliating in your grade based on the fact that you were personally revolted by me. Are there other questions? So, all right, now let's see if I can find the list. Okay, so with that being the case, let us start by thinking about the short and easy nature paper that is Begun and Aquadro. I know I must have it open somewhere. And let me see if I can share my screen. Let's see. Are you guys looking at the Begun and Aquadro paper? And is Sebastian here? I can't actually see who's here yet. Is he here or is he still working?
Yeah, I'm back.
Back, man. Good. Did your exams, you survived your exams okay?
Yeah, it went better than expected. So, Carmen, Mitchell, Brooke, and Marisa, I bet you are gonna kill the exam when it's your turn.
Okay, good. Excellent. So, have you had a chance to look at this paper yet?
Yeah, this one is quite interesting. We like in our lab we have the opportunity to collaborate with Chip Aquadro, and like he's amazing. He's like genius level.
Yeah. So
amazing. And this paper, like how it made me think about mosquitoes as like any Drosophila paper, just because of like how they like remind you about like the patterns of bands in like the polyethylene chromosomes, and how like you can relate this to like stable areas. And this is like what they kind of show here, like they show like highly divergent areas with low recombination rates.
Yeah.
But they kind of like blame all that on
Yeah.
gene hitchhiking, right?
Yeah, we'll talk about how we'll talk about their explanation versus their observation in a minute. Yeah, and if any of you want to know sort of a personal story, for those of you, so for years I've taught this class with Mike Zwick, who's become too much of a dean and too much of an important person to have time for this. But if any of you sufficiently hate Mike Zwick, you can blame Chip Aquadro. So Chip was a very young faculty member who discovered an even younger undergraduate, Mike Zwick, when he was a freshman at Cornell, and sucked him into the lab originally to largely wash dishes, which is what he did for the first year or two of his undergraduate career, before he went on to follow Chip and become at first a Drosophila geneticist and then human geneticist. So much of this research actually, I think, was happening when Mike was an undergrad in Chip's lab. And but Dave Begun and Chip Aquadro were of course, he was washing dishes. But anyway, we all come from somewhere. So Sebastian, tell me what's going on in table one. What are those different columns and what is, like tell me what each of those columns are.
Okay, so first the column that says like the gene region for each chromosome, the gene region they like included in their analysis, they assessed.
Yep.
The coefficient of exchange, like I didn't quite get it. Like I get nucleotide diversity, which is the next column, divergence between the samples.
So it turns out you're less than 100 years old. And coefficient of exchange was the term coined by, I believe, Sturdivant in 1910 to mean observable recombination between markers. So the coefficient of exchange is the rate at which I can observe an apparent recombination between linked markers. And so this coefficient of exchange is an estimated recombination rate in that region.
Okay.
So that is,
Okay.
we have what you and I and anyone from even the late 20th century would have simply called recombination rate or estimated recombination rate. They've been very precise and called coefficient of exchange to remind you that this has been estimated from some set of markers they can observe in that region. But you should think of this as recombination rate.
Okay, and I just have a question from this, like from this table. So the regions they like are mostly focused on are like this region from the chromosome 1 and the chromosome 3, right?
And 2, there's a bunch from 2 also in there.
Okay.
So, oh yeah, for those of you who are not fly geneticists, flies have at most 4 chromosomes, but the 4th one has no genes on it, really. Chromosome 1 is generally called the X chromosome, and it is the chromosome that is associated with sex in flies. Chromosome 2 and 3 are the autosomes. So basically, your chromosomes in flies are X, 2, and 3. The size of the X is about half the size of 2 or 3. And of course, it's this sort of, diploid in females, haploid in males. So we have, a whole bunch of loci on the X, yellow, white, notch, vermilion, forked. We have a whole bunch of loci in chromosome 2, ADH, DDC, the amylase gene. We got a whole bunch of loci in chromosome 3, the esterase 6 locus, rosy, heat shock. Oh, by God, they even have a chromosome 4 locus with no estimated chromosome 4 doesn't recombine, by the way, with no estimated recombination, no estimated, remind me, Sebastian, what is pi again?
Pi is the nucleotide diversity.
Exactly. Tajima's estimator of theta. You guys know how to calculate that. You have done that. You did that extensively on like homework 5 or 6. And what does divergence refer to here?
I think, is this the divergence between the species of Drosophila for this like same marker?
Exactly. So if we look at the yellow acute scute region, if you pick 2 flies at random in melanogaster and you ask what is the probability a randomly picked nucleotide will be a heterozygote, you're going to get something that on the order of 0.001 for yellow acute scute, 0.003 for PGD, 0.004 for zeta, almost 0.1 for white. That's the nucleotide, that's sort of heterozygosity. The probability 2 randomly picked nucleotides are different. And divergence is again the same thing, but not within melanogaster, but between a randomly picked melanogaster and a randomly picked simulans. So here, if you pick a nucleotide in melanogaster and you pick a nucleotide in simulans, there's about a 5.4% chance they differ if you're looking at yellow acute scute. There's about a 0.5%, there's about a 5% chance they differ if you look at the period locus, and almost a 5% if you look at vermilion, almost 5% at ADH, 7% at whatever that metallothionein A gene is, 5% on the chromosome 4 locus. So if you pick any 2 nucleotides in between melanogaster and simulans, you will shockingly see, almost no matter what the locus is, the amount of divergence, the probability those 2 nucleotides differ, is about 5% between mel and sim. And that's because there's about a 2.5% divergence since the common ancestor of both mel and sim, sort of in each lineage. These numbers hold up very well. This is, if we went to the whole genome paper from 10, 15 years ago, 10, 12 years ago now, you will see that these numbers are not dramatically different. That Begun and Aquadro did a pretty good job of estimating divergence at these handful of loci, and these handful of loci are not atypical of the genome. And many of them are Dave and Chip sequencing directly, and some of them were taken from the literature for both heterozygosity and for divergence. Does anyone have any questions about what the data is and what's being shown in table 1? So, Carmen.
I have a question about divergence really quick.
Yep.
So I think I looked it up online just to get a definition, and then it was basically saying like, oh, it's the number of like unique mutations that have emerged between the two species. That's not really. It seems like what you said though is it's not that they're unique. It's just that they happen to be different when you draw them, I guess.
Yes. Yes. So that is a field unto itself. So it's very easy for me to align sequences and count the number of differences and report the number of differences divided by the total number of sites I looked at. That may or may not be a good estimate of the total number of differences that have occurred in the history of the two species if some sites have had multiple mutations at them. So if a site has mutated once and then mutated back again, we wouldn't be able to see it this way. Or if a site has mutated once and then mutated to a totally different site later on, we might only be counting it once rather than twice. So from almost the beginning of molecular evolution, there have been things called, what do you call those? Oh, I lost the word for it. Anyway, if you assume some model of molecular evolution, you can, of course, estimate the true number of mutations rather than the observed. They're called correction factors. That's what they call them. And rather than using the observed number of mutations, you can correct it under some model of evolution to what you think the true value is. And if you have enough sense of these things, that if you assume the underlying process is something like a Poisson, if the value of divergence is small, it's going to make almost no difference whether you take the observed value or you attempt to correct it. And if the value is large, correcting it is probably going to introduce, it's going to introduce a lot of variance. And if your model is in any sense wrong, the amount of variance it introduces is probably worse than what your starting values were. So using correction factors is often a dangerous thing unless you're really, really, really confident of the underlying model. But here, I think they've just used, they're just used the number of differences divided by the length. Questions?
Thank you.
And divergence here is exactly the same thing we were calculating in the McDonald-Kreitman paper, where instead of doing it as a percentage, they did it as counts. And you saw how those counts were made in McDonald-Kreitman, literally by eye practically in this era.
I have a question,
Yep.
just, I guess, about the values. Are these, so 5% of the time you're selecting something that would be the same, right?
Mm-hmm. No, 5% difference. 95% of all sites are identical at yellow or key.
Right. Okay. So basically, is that would that be considered like a pretty closely related, or is that kind of saying that
right.
these are far apart?
So you and I, assuming you're not a Drosophila geneticist, if I were to put a plate in front of you that was half melanogaster and half simulans flies, neither you nor I would be able to tell you which ones were melanogaster and which ones were simulans. To us, God damn it, they're just little flies. They look identical to each other. This was a classical crypto species where people didn't even know there were different species until they tried to, until they started making plates of them and realized some of them will only breed with some and others will only breed with others. And then when people started really looking closely, they started seeing actual differences in the phenotype, sort of observable differences between simulants and melanogaster. Dave Begun will never, ever mistake a simulants from melanogaster. At a glance, he can tell you which is which. But you and I would struggle with that. So most people think melanogaster, so as a rule, you'd think melanogaster and simulants are really, really, really close sibling species. They're as close phenotypically as just about anything in nature you would call two different species. And this amount of divergence, 2 to 5%, is typical of things we call sibling species, unless they happen to be large charismatic mammals. It turns out humans are really good, probably, at distinguishing small, tiny phenotypic differences in large mammals. So the divergence between you and a chimp is like 1%. And there is no human on Earth that would ever mistake a human for a chimp or vice versa. The amount of divergence between, like, I think maybe even between a tiger and a house cat may only be 1 or 2%. So most of you can tell the difference between a tiger and a domestic cat without very much difficulty at all. And it's not just by size, sort of you see all sorts of face shape differences, paws look different, whatever. But for organisms that humans are not great at discerning the phenotype of, 2%, 5% divergence would usually be something that we'd struggle to say they are different species without doing molecular genetics or some deep biological observation of them. Okay. Anybody else got questions about this data? Okay, Carmen, let's look at figure 1. What is figure 1 showing us?
Can you scroll up a little bit? Oh,
Yeah,
there we
you
go.
bet. Can you see it now?
So it plots coefficient of exchange on the x-axis and the nucleotide diversity on the y. And it basically shows us that as recombination rate increases, nucleotide diversity increases.
Perfect. Everybody convinced by that relationship? Looks more or less believable. At the time, some people were a little concerned that some of this regression was being driven by a few of the points in the sort of highest recombination region, that it kind of looked maybe if you pulled these 2 points out, this regression isn't quite so significant. But having said that, this observation has been repeated 1,000,000 times since then. It is true over and over. It is true over the entire melanogaster genome. It is true in many, many other species. It's true in humans. It's true in tomatoes. It's true almost wherever you look, that in regions of low recombination, there tends to be very low diversity. In regions of very high recombination, there tends to be higher diversity. And there is something resembling roughly kind of sort of a linear relationship. Although most of you would probably believe I could plot other functions in there that is increasing, but not necessarily linear to explain it. But that relationship, while first noted by Begun and Aquadro, is indisputable in virtually every organism on Earth. Does anyone have any questions about this? So, Zhangji, what is going on in figure 2?
Okay, so figure 2 is plotting effective or estimated recombination rate on the x-axis and divergence on the y-axis. And in this case, the authors claim that there is no correlation because it is a flat line, basically.
Yep. And this is pretty much true too. That I think when you get to huge sample sizes and whole genome type data, you can start seeing a very, very weak effect of increasing recombination, leading to slightly higher divergences, maybe. But grossly, you will see this pattern over and over again, that grossly, there is very little evidence for any recombination effect on divergence. And Begun and Aquadro were probably the first to point that out carefully. And this observation holds really at genome scales as well. Does anyone have any questions about figure 2?
Actually, I have questions about figure 1 and 2. So in the caption for both figures, there's a correction for the X-linked genes and genes on the autosomal genes, and I really think that they should have done separate regressions for these two types of genes instead of making them, instead of doing the correction.
Yep. You can, they've colored them differently. You can hardly see the color. That the ones that look like full circles are like autosomes, and the ones that look like kind of hatchy circles, if you put your face like right up to the computer, are X, or vice versa. At this scale, they sort of look the same. I mean, they both look like they're trending the same way. And they probably had no statistical power to separate them. You're absolutely right. These corrections are very, very hokey. They're very, sort of very rough. And if you were all concerned about this, you should certainly treat the X and the autosomes differently. You may even want to treat every chromosome differently from one another. But it turns out these relationships hold within every chromosome now that we have a lot more data, but you're totally right. And the reason we're making these stupid corrections is for the obvious reasons. The X chromosome, they're on the autosomes, both boys and girls both are diploid at every autosomal locus, males and females. For the X, females are diploid, males are haploid. So fundamentally, there are 3/4 as many X-linked alleles in a population as there are autosomal alleles. So they've made an adjustment to nucleotide diversity where we're recognizing that for the autosomes, we're really estimating 4N mu, whereas for the X-linked loci, we're estimating 3N mu, because they have sort of a different population size. And the other thing many of you might not know about Drosophila is there is no recombination in boys. Male melanogaster do not recombine. So for the autosomes, the recombination rate is proportional to the number of female chromosomes. And for the X, the recombination rate is proportional to the number of female chromosomes. So for the autosomes, effectively half the chromosomes get a recombination every generation, whereas for the X, 2/3 of them get a recombination every generation because 2/3 of them are sitting in girls. So they have adjusted the estimates of recombination rate and the estimates of nucleotide diversity to try to put them all on the same scale. And a much simpler procedure might have been just to ignore all of this and leave X on X scale and autosomes on autosome scale. Actually, that would be a much cleaner way of doing the analysis. But given that they only had probably 10 or 15 well, for the X, it looks like they only got about like 6 or 7 loci, that they probably just didn't have the power to do it that way. But with the whole genome in front of us today, we'd have taken your suggestion.
I've kind of, I don't know if it's a question or really a comment, but maybe just thinking through like what experimentally you could do. Because for this today, I can't remember if they really talked about whether or not these recombination observations were related to like on the chromosome themselves or if it was across chromosomes. Because I think if you're then taking out X and you're not measuring those, you almost it almost necessitates that you have to include multiple chromosomes in an analysis. But if we're just looking at the recombination events that are happening like on different sites of a chromosome, then you could separate out the two.
Yeah.
Autosomal analysis and sex linked, I would think.
So all of these recombination experiments are were very, very painful to do at the time. They have some kind of flanking markers around the locus that they can measure or observe in some fashion. For some of them, they were actually visible markers. In other cases, they were more like RFLP type markers. And these recombination rate estimates were in very large sample sizes, counting the number of times they observed exchange in the flanking markers surrounding the region. And some of these experiments were focused on a single locus. Like I'd be surprised, I don't know what that 13 citation is, but it's very likely to have been a dissection of the white locus, carefully looking at recombination rates. Whereas others of these may have been trying to estimate the recombination rate along an entire chromosome with many visible markers. And that's why some of them are probably the same citation multiple times, whereas others of them are sort of just one citation that tried to take apart that one locus. But it totally happened in a lab. It totally happened in a lab that may or may not have been wildly affected with transposable elements, which we're only beginning to wrap our heads around in the 80s. These nucleotide diversity estimates are probably really solid. The coefficient of exchange estimates you might have worried about, although I think all of them have just become more and more solid over time. That a lot of these large fly populations probably made up for a lot of the otherwise inherent noise in these experiments. I don't know if that got at your concern, but.
I think so. Yeah. I was just thinking about how they were really measuring like what was the origin of the recombination really?
Yeah, it is literally, I've got a bunch of flanking markers. I've got a bunch of males with we're going to call them plus markers across the chromosome. I've got females with a bunch of minus markers across the chromosome. I created an F1, which has, it's 50-50 pluses and minuses, and then I created an F2, and I'm looking for the distributions of where the recombinations occurred in those F1s. And if I'm on the X, I'm going to be careful about which sex is which. And well, in general, I'm going to have to be careful because recombinations only happen in females. So these were, if there were visible markers, it was easy to get large sample sizes. If there were molecular markers, it was considerably harder in the serum. Questions? Okay, so we have, this is a great Nature paper, one table that's the data, and two figures that are the entire analysis of the data, both of which are just a linear regression of, in both cases, recombination rate, once against nucleotide diversity, once against divergence. And we see a pattern where there is definitely, well, a convincing correlation between increasing recombination and increasing diversity, and no relationship whatsoever between recombination and divergence. So, Jim, why did I do two different, why have I done both of these experiments? What am I trying to distinguish here, if anything? Why am I seeing different patterns?
I mean, I think this might be wrong, but does it really just get to the within population or across population question?
Tell me about if the whole world were completely neutral, what would I expect the relationship between nucleotide diversity and divergence to look like?
Not following entirely.
So let's go back to homework 3 or 4. Before we did any before natural selection came about, maybe homework 2 or 3, and we're reading the McDonald-Kreitman paper or something like that, and I'm estimating nucleotide diversity within populations, which ought to be, which is, you know, an estimate of theta. And I'm looking at divergence between populations. What is divergence between populations proportional to if the whole world is neutral?
I guess it should be the same, right?
Yeah, exactly.
There shouldn't be any difference.
Yeah, so if the world is neutral, divergence is a measure of mu-t, of the mutation rate times the time between the species. We did those sorts of problems, right? Where you were given divergence between the two species, you were given nucleotide diversity, and you had to find the population size. And the reason that worked is if the world is neutral, divergence is simply mu-t, and nucleotide diversity is 4n mu. So if I had done the plot of divergence versus nucleotide diversity, I would have expected a completely flat line comparing divergence and nucleotide diversity. And because in each case, the value of the point is just something like time divided by 4n, or 4n divided by time, depending on which way around I plotted it. And it just reflects the mutation rate, that you're going to get an estimate of the underlying mutation rate. And so if I plotted these two against each other, I expect a flat line. Instead, one of these two things is increasing with respect to the coefficient of exchange, and the other thing is staying completely flat with respect to the coefficient of exchange. So that suggests that the whole world isn't neutral. If the whole world were, we would expect both nucleotide diversity and divergence to simply be estimating the mutation rate. One of them is not doing that. And the conjecture of the authors is that divergence is reflecting the mutation rate, that the reason divergence is completely flat across the coefficient of exchange is all of these have fundamentally the same mutation rate. And the reason, and we're estimating neutral divergence here, but here with nucleotide diversity, with heterozygosity, we are seeing the effects of selection. And their conjecture is linked selection. It's not necessarily selection on each and every site, but linked selection is much more effective in regions of low recombination than in high recombination. So the effect of selection is stronger here than it is here. And selection is reducing diversity more here than it is here. And given how often this picture is repeated in species after species, it seems very likely this interpretation is correct, that linked selection is leading to lower nucleotide diversity in regions of low recombination than in regions of high recombination. Now, the authors also make some conjecture that the type of selection, the sign of the selective effects, is positive. It's rare alleles that are increasing in frequency and going to fixation, that are sweeping through the population because they are good. That's possible. But linked negative selection has the exact same effect. If most mutations are bad for you and being driven out of the population because they harm the organism, the observable effect is the same. You will have lower diversity in regions of low recombination and higher diversities in regions of higher recombination. When you call it, when the average effect of an allele is negative, bad for you, minus S, for historical reasons, we call that background selection. That's a term coined, I believe, by the Charlesworths. And when the sign of selection is positive, we call it selective sweeps. That term was coined by, I think, Maynard Smith. But mathematically, they have the same effect. They both lower diversity in regions of low recombination, and you lead to higher diversity in regions of high recombination. And neither background selection nor selective sweeps nor anything affects the rate at which neutral sites diverge. So that's also provable. It's maybe not obvious, but it's a relatively straightforward proof that selection going on at a small number of sites does not affect the rate at which neutral sites fix between populations. So the usual interpretation of divergence is most of the sites you see that are diverged are probably neutral fixations, and most of the sites you see in a locus might be neutral, but the total diversity is probably being affected by neighboring selected sites. Either positive, in which case we'll call them selective sweeps, slash, what's Gillespie's term for it? Genetic draft, or negatively, background selection. Do we have questions about this?
I have a question about figure 2, which maybe is kind of a reach. But I was looking at, so you have a different kind of variance around that line when coefficient of exchange is low versus when it's high. Like from that information alone, could you like make any, like, I guess, leaps as to whether or not a sweep happened, or
Yeah. So
maybe not, I don't know.
So you're probably right that you might be able to tell the difference between background selection and sweeps by, say, the pattern of the, sort of the, almost like the correlation structure of pi moving across a chromosome. That A selective sweep probably results in the spot that was actually selected having the lowest diversity, in regions moving away from it, sort of slowly increasing in diversity. And if the sweep were strong and recent, you might see a pattern of near zero diversity for some period, and that sort of fall away with the recombination rate. Whereas if this were constant low-level background selection, you might see no particular autocorrelation structure in pi, say. So you might imagine you could develop tests to try to distinguish positive and negative selection by looking at, say, autocorrelation functions in heterozygosity across chromosomes, or put more simply, in haplotype length type statistics. But a haplotype length you might reasonably think of as an autocorrelation in pi. So there are tests, they tend to be weak, that try to look at that try to find positively selected regions from its pattern of autocorrelation in pi, or haplotype length, when we sort of say it most simply. Anyone have other questions or comments? Okay, that's begun in the Nope, sorry.
So I have what might be deemed as a really dumb question. So for table one, I'm just curious about like how some population genetics and evolution papers are shown.
Yep.
Like how they show their data, just like they might seem just as point estimates, and we know like how like they fit into this like dynamics we're understanding as we're taking this class. But I'm just curious, like why don't we have like intervals and just certainty about like some of these estimates?
So so why don't we have confidence intervals on any of these estimates? Honestly, because many of the people who did these estimates were probably not strong enough statisticians to think about a variance as well as a mean. The vast majority of these estimates are simply first moment estimators. They're an estimate of a mean. And many of the people doing it, if you look at the papers, they are astonishingly strong fly geneticists who are decent statisticians, but not great. They are the Chuck Langleys of the world who have amazing intuition, but won't calculate a variance for you most of the time. And so I bet if we went back to many of the underlying cited papers that have the data here, there would be enough data in them for us to calculate both a point estimate and an uncertainty in it. And we could probably even think about weighting some of these by their uncertainty. But for the field as a whole in the 60s and 70s, that was not widely done. And also, you can sort of see it too in a bit before the existence of a hand calculator, even calculating means could be a challenge. Never mind calculating variances. And these are, Begun and Aquadro are certainly after calculators exist, and even after spreadsheets exist. But the underlying data from some of the other papers may even be older than that, and can reasonably date to eras when it was a challenge even to count up some of these things accurately. Other questions? Okay. So if you have any doubt whether these patterns are true, read the whole genome melanogaster sequencing paper, whose first author is either Chuck Langley or Dave Begun, or vice versa. It's either, I can't remember if Dave is first and Chuck's last, or vice versa. But anyway, go read the whole genome sequencing paper, and you will see a lot more of this, and you will see that that Begun and Aquadro got it largely correct. I mean, it's just right. Other questions about this right now? Before we change gears entirely and head into quantitative genetics. So let me see if in here, instead of, oh, there are people in the chat. Oh, okay.
So sorry, that was just
Yes.
me saying stupid
Yeah.
stuff.
Is the same graph in Gillespie's book? I wouldn't be surprised if it was. I don't actually know that, but it would almost surprise me if John didn't put it in his book somewhere. He may have also faked the data and like given you a sketch of it. Okay, so we are moving now to where am I? Share screen. Barton and Turelli. This one was long, painful, maybe interesting places. So we actually have to pop my notes for the questions. So I'm going to approach this paper two ways. I'm going to go through a lot of it and ask you guys specific questions about what they're talking about in specific sentences. And hopefully in that fashion, we'll come to understand what Barton and Turelli are talking about. But if I don't highlight the right sentences and you guys still have like deep, like what the hell does that mean kind of questions, please shout them out as we go along, because I've hit some, but maybe didn't hit all the right ones, and we can sort of talk through them as we get to them. But so we're going to start with Kiana on page 338, and a sentence that reads, where is it? Despite Fisher's reconciliation of Mendelism with the resemblance between relatives. What does this sentence mean? Is Kiana on? Yeah. No?
Yeah, I'm here. I'm just processing, preparing.
What was Fisher's reconciliation of Mendelism with the resemblance between relatives? Do you know what that means?
Well, Mendelian inheritance necessarily, or is that the resemblance among relatives for traits that are polygenic can be governed by a lot of individual genes that are inherited in a Mendelian fashion, but this is my guess. But for quantitative traits, the inheritance of the trait itself doesn't necessarily follow a Mendelian pattern.
Yeah. So site 79, if you had looked it up, would have said Fisher 1918, and the site could also have simply been your homework 9. Fisher's reconciliation of Mendelism with the resemblance between relatives is what you guys did in homework 9. In homework 9, you showed that if you have loci that are inherited in a Mendelian fashion, you have a big A and a little a, you have heterozygotes, you got homozygotes, and that the two alleles have different effects, and you have a lot of loci, you can predict. Here, the word resemblance means technically correlation or covariance, that you can predict the covariance and phenotype between relatives from simple Mendelian genetics. And Fisher 1918 was the first thing that showed that, and it is your homework 9. So whenever they're using terms like resemblance between relatives or correlation, they're talking about the things you derived in homework 9, and Fisher was the first person who did it. And this matters because, well, it doesn't matter. Well, I mean, we're talking about history here, but there was, so starting in about 1870, almost 1880, a guy by the name of Galton had a model of inheritance that was the only model any smart biologist believed. And that model of inheritance had created, traits were created by blending the phenotype of mom and dad. And in fact, they were created by blending the phenotype of all of mom's ancestors and all of dad's ancestors into the kid. Galton and Pearson derived all of those correlation equations with a model of inheritance that was fundamentally this weird non-Markovian blending kind of model. The model is utterly wrong and almost impossible to understand from our sort of perspective of knowing how inheritance works. But the entire field of quantitative genetics developed most of their major results in an era when they thought that inheritance worked by this weird blending pattern. And when Mendelianism was first discovered in like 1904, 1905, or rediscovered, most serious biologists thought Mendelian genetics was like this weird little plant thing and had nothing to do with anything important, because every trait that we care about, I can describe using the regression equations that you guys did in homeworks 9 and 10. That from a practical standpoint, I can understand the world completely using what you guys did in homework 10, which doesn't require anything about Mendelian transmission. In fact, at first, the model that they used to drive all that was anti-Mendelian. And it was until 1918 when Fisher first recognized that the correlation between relatives could actually you'd get the exact same results if from you know, a Mendelian model that we would now accept as correct. And the second half of that sentence, the standard equations for the response of phenotypic means to selection are essentially pre-Mendelian, means R equals H squared S, that Lush's equation, the response of phenotypic means to selection are all pre-Mendelian, that all of this stuff worked and was used and useful long before anyone understood how genetics actually happens. And so that there is this, the field of quantitative genetics has this tradition of having results that without really even needing to know anything about genetics. And in fact, getting useful and correctly usable tools with a fundamentally deep misunderstanding of how genetics works. And
I'm not the most well-versed in the history of evolutionary quantitative genetics, but I'm wondering, is this moment pre-modern synthesis, post? Is this the modern synthesis?
This is the moment the modern synthesis began.
Okay, cool.
So,
I just wanted to place it in context.
Yeah, so Fisher 1918 is the cause of the modern synthesis. It is the moment when we recognize that everything we know about quantitative traits might be explained by what seems to be the truth about genetics, which is coming out of the Sturdivant lab, that we are learning how genetics works, and it is consistent with everything we know about phenotypes. And that's the moment the modern synthesis begins. So the way to people often talk about the modern synthesis is the theory of it was largely attributable to Fisher, Wright, and Haldane, where Fisher first establishes that Mendelian genetics is consistent with the largest class of observations we're going to get. And then Fisher, Wright, and Haldane start doing the mathematics for what happens when natural selection exists or doesn't exist. And those were a lot of the results we did in homework 5 and 6, were sort of early Haldane and Wright and Fisher kind of results. And once people understood that natural selection could push around allele frequencies and that genetic drift exists and all that other good stuff, serious biologists like Jablonski, Meyer, those guys started using what we knew about the way selection ought to work and the way genetics does work to understand what they're seeing in nature with their favorite species. And, you know, Meyer showed that like all this stuff is consistent with everything he knows about birds. Stebbins showed that most of this shit is consistent with everything he knows about plants. G.G. Simpson started showing that much of this stuff is very consistent with the fossil record, and so forth. Jablonski, all this stuff is completely consistent with experiments in flies. And the modern synthesis as we know it sort of develops between 1918 and we'll say Simpson's book, maybe 1948, 49. I don't remember when Simpson's book came out. Yeah, that's the rough view here.
Can I follow up that on a question of,
Yes.
pardon the ignorance or if I missed this in some reading or part of the reading, but I've never heard of this time or this historical period, the modern synthesis.
Yeah.
It sounds like you kind of just answered what it is, but maybe in one sentence,
Yeah.
what is it?
So the modern synthesis it's a term probably coined in the 50s to describe the unification of Darwinian thoughts about natural selection, Mendel's observations about the way genetics works, and everything we know about the phenotype of organisms in nature. It was largely showing that we could understand things, almost everything in nature could be understood as being the result of the way in which we think genetics works, the way in which we think natural selection ought to work, and what we can see in nature. The modern synthesis is what gives us biology as you understand it, rather than a set of unrelated fields that might have been called botany or lepidoptery or, you know, animal husbandry or microbiology. That the modern synthesis suggested that all life on Earth could be understood in roughly the same way as being the result of Mendelian genetics, natural selection, and yeah, basically those together.
Gotcha, thank you.
Yep. So the beginning of this paper is telling us that 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 quantitative genetics predates any understanding of how actual inheritance works, and that you could have done homework 10 without understanding anything about genetics. But homework 9, Fisher's reconciliation of Mendelism with resemblance between relatives, is what unifies how genetics actually works with things we can measure in nature, like the correlation between relatives, or how much the offspring phenotype will differ as a function of the parental differences between means. So, Michelle, on the next page, there is a line that begins most continuously varying characters. Right here in the genetics of quantitative variation. It says most continuously varying characters in most populations will respond to directional selection. What does he mean? What does this mean?
What does he mean? So continuously varying characters would be like non-discrete.
Yep.
And they will respond to directional selection. So if selection is moving like the mean of this trait in one direction, it can respond. So if you want to make like cows that are bigger, you can select for that and they will respond to it.
Exactly. The sentence says R equals H squared S almost any time anyone's ever tried to do it. The experiment almost always works. That if you select for parents having a bigger mean, your kids will have a bigger mean. If you select for parents having a smaller mean, kids will have a smaller mean. That this is not, this happens over and over and over again. In fact, if you try to do an experiment where this doesn't happen, where you can't get a response to selection, you'll almost get a famous paper, because it is so rare that that you don't get a response to selection. It'd be noteworthy. And to the extent that there is any rule at all for when you don't get a response to selection, it's you can't select for fluctuating body asymmetry. You can't select for say systematic differences that vary from one generation to the next in how asymmetric a body plan is. You can select for an asymmetry sometimes, but you can't make it go back and forth in a non-random way. And in fact, you often have trouble just selecting for asymmetries overall. Body plans tend to be really, really, really canalized in most species, and you can't make them. You can't get a huge arm and a little arm very easily, usually. But most everything else, these sort of very famous papers where a guy by the name of Ken Weber started to ask the question, really, is there anything I can't select on? And he used to do these experiments where he'd blow up pictures of fly wings. He'd have a little camera set up and look at the fly wing and blow up this picture of the wings. And he'd pick like a set of veins in the wings. And he'd ask, can I select for the angle made by the intersection of these two veins to change? The answer is yes. Can I make one of the veins bigger than the other? Yes. Can I do anything? And it seems like when you try to do experiments, there's almost no phenotypic character you cannot make move in the direction you ask it to move, if you do. And you certainly know that for anything that you eat, pretty much has undergone 150 years of hard selection. If you are, certainly if you are a meat eater, cows and pigs and chickens really resemble nothing like anything in nature practically. And for vegetarians, it's even worse, really. I mean, the corn, I mean, a few thousand generations ago, it looked more like grass, more like wheat than what you would consider corn. Almost none of the vegetables you'd find at a grocery store today pretty much even existed 200 years ago. We've just pushed all that stuff around dramatically so that they, taste good, basically. Survive the trip to.
Dave, I was gonna interject. I was reading something about flaxseed and how flaxseed was selected for like plumper seeds to get more oil out of them. But they're actually really hard to find flaxseed, I guess, when it disperses the actual seeds. So humans have selected for ones that don't open up their pods so that they can find them easier. And it's like kind of crazy to think about. I think, yeah.
Exactly. There's an enormous number of things we eat that the plants that can't actually breed on their own, that need a human to like physically cut a little part and add a little part here, do a little. It's not even like it's not even a viable organism on its, sort of a reproductive organism on its own without a little bit of human interference. And we have found that we can select for almost anything we want, and have done so to make things easier to farm, to make things taste better, to make seeds not disperse into our neighbor's yard as much, to make them disperse extra. We've selected everything. We've done all of that good stuff.
Can I ask a question?
Yep.
I think it's in the same line. So I've kind of, in like research presentations, sometimes people talk about this concept of like evolvability, or like how organisms might have evolved to be evolvable. Is this related to that? Or can you like sort of explain what that means, I guess?
Sure. Let's talk a little bit more about it as we get a little further into this. But we will encounter in a very short while notions asking questions about how many genes are involved, what is the mutation rate, and what is the average effect of a new mutation. That you can imagine your heritability, fundamentally. So evolvability, R equals H squared S. Evolvability is the size of R. So easily selection can be any value of S you'd like it to be. So evolvability in this context really means H squared. And really it means, in the context you're asking, H squared in the presence of strong selection over extended periods of time. So those questions will be asking, what maintains H squared bigger than 0, even if selection is strong for long periods? And in order for H squared to remain bigger than 0 over significant periods, it'll probably turn out to be a function of the number of genes involved, the mutation rate of those genes, and the average effect of new variants. And we'll probably think about some of those things in terms of something we might call a mutational variance, and then later just overall heritability. Does that make sense?
So does evolvability have anything to do with, because they mentioned in the paper, like when you select for something, the population will adopt that trait, but then after you stop selecting for it, it will kind of go back to its old mean, I guess. Yep. Does evolvability have to do with that ability to like go back to where it was, presumably to adapt to some, environmental change or something that might be like transient?
Yeah, it could. Most of the time, evolvability will simply mean the existence of additive variance for that trait, or the existence of additive variance that I am able to select for. So it may, we'll see. Like, if it turns out that traits are highly pleiotropic, and that although I still have additive viability, I still have VA is still non-zero and significant. If it turns out the same alleles that contribute to the additive variants at the thing I'm trying to select for also contribute to variants at other phenotypes, some of which might be very bad for the organism, I may find that I cannot evolve even in the presence of significant VA due to pleiotropic effects and selection going in opposite directions. And in fact, the whole ability to evolve might actually be a function not just of VA for my trait, but of the entire variance covariance matrix of VA, the genetic covariance matrix, and all the different selection gradients on it. And for a single locus, it's just really asking whether you still get VA. But in a whole organism, it's asking whether the whole variance covariance matrix is such that you can still move in that direction without pleiotropic effects stopping you. And we'll see some of these discussions. Although they won't use the word evolvability, they will use words about those things. So there's a key idea in this little paragraph that's just jumped over, but is probably important for all the biologists in here to see really well. And Brooke, there are two sentences that start in this paragraph that begin some selection experiments and end with a bunch of citations after fossil record. And what is in these two sentences with a bunch of citations? What are we learning here? You're still with us, Brooke, right? Yeah.
She's on mute.
Yep, sorry, I was on mute and I was reading it
goodness.
out loud to myself. Oh my goodness.
Yeah.
Love it. Love it. Good morning. Okay, so basically we're talking about how there's this altered mean that is 10 times the initial phenotypic standard deviation over time, which is kind of an unprecedented rate seen in the fossil record. And I think when I was reading this, I was thinking a little bit about like how much we understand about rates of change that are occurring in existing populations versus those that are either extinct or ancient, older populations. I wonder if this is kind of getting into this sentence is really talking about that idea of microevolution versus macroevolution. So changes that are occurring over generations of time versus, I guess, the record that we have of speciation from fossils.
Yeah. So the first sentence, I can do experiments where I push the mean of a population 10 standard deviations in 100 generations, like 10 standard deviations, what out, that's like, what's a standard deviation for height for humans? Probably maybe like 5 inches. So that'd be like pushing humans to be like 50 inches taller within 10 generations. We're now like all at like 9 feet tall. You can make enormous differences in these artificial selection experiments in roughly the blink of an eye, 100 generations, that there seems to be nothing that would stop us in these sort of artificial selection experiments from moving a population mean all over the place, just way, way different than what it is now, if selection wanted it to go that way. But if you look at anything like a fossil record, what you see is you can recognize the same fossil, usually if you have a good record of it, for hundreds of thousands or millions of years. So that if you have a good fossil record, you will often see it looks like a population mean hardly changes for hundreds of thousands or maybe even millions of years. And in fact, when it changes a standard deviation or two, we often call it a different species. So we'll be looking at a fossil that looks like it barely changes, and then, we see another fossil that's 2 or 3 standard deviations away. We'll often call that a different species. And we'll say, looks like there's a speciation event. Those speciation events that occur in the fossil record over hundreds of thousands of years could have occurred in like a couple hundred years in real life if selection had been strong and wanted it to happen. If selection had acted to make that happen, it could have happened in the blink of an eye. And there is literally nothing you will ever observe in the fossil record that is a change that is too fast for quantitative genetics to have explained. In fact, there is a series of papers, by Turelli and Gillespie, among others, that if you assume neutrality, if you assume there is no such thing as stabilizing selection, the fossil record doesn't change nearly enough. You should be seeing far greater changes in the fossil record if the world were just genetic drift. Means should move about a lot more. And the fact that you can recognize the same fossil species over and over and over again is sort of your proof that it is probable that natural selection was not acting to move it in any direction. I mean, it's certain. And it was not even neutral. It's probably that it was under some form of selection that kept the mean where it belonged, that was a form of stabilizing selection, which we'll talk about a little bit more later on. But if you ever run into a paleontologist that says, look, I have these 2 species, I have these 2 bones, and they differ, they found them, they're only 30,000 years apart. And they're 3 standard deviations different in size. So what? That's the answer. Great, congratulations. Look, that happened. There is nothing surprising about that. Like, there is no reason, really, really weak selection couldn't have taken care of that for you. And that's what that paragraph in those 15 citations are sitting there showing you, or alluding to is probably a better word for it. So, Young Xian, on the next page, we're going to see the word for the first time, the infinitesimal model. What do they mean by? And we're going to see actually several phrases of art in this sentence that begins on page 340 with first the word infinitesimal model, then genetic additive variance and Gaussian distribution of breeding values. So what is meant by any of this nonsense?
So by saying infinitesimal model, what I understood was that the effect of each alleles are very, very, very small. So we can,
And therefore what?
And therefore we can maybe not consider the Mendelian.
If the effects of individual loci are small, how many loci must there be?
There should be a lot of loci. Yeah.
And so the infinitesimal model is effectively, again, what we were doing in homework 9, where it is every locus contributes only a small amount of variance. And there are a lot of loci that might contribute that variance, that much of the variance is additive, and there are so many loci that breeding values, by that we mean the additive variance contributed by any individual, breeding value and additive variance are almost synonymous terms. That the additive effects or the total additive contribution from an individual is approximately normally distributed. Gaussian. The word Gaussian, of course, and normal are synonymous with each other, and we'll go back and forth between them. So that the standard model that we will have in our mind, often, always, really, is there are a lot of loci. Those loci individually contributed relatively little variance. Together, they might contribute quite a bit, and that both the phenotype at large and the additive variance contributed by sort of the genetics are both Gaussian distributed. That'll be the assumption for a lot of this stuff. And so Mitchell, Wright-Castle estimation. What are we doing with that stuff? The widely used Wright-Castle formula estimates something. What is it estimating?
The number of effective number of loci.
Yeah. So that's it. So starting in this is a great story, right? So starting in about when was Wright-Castle estimation? Good Lord. It's before the synthesis, it's before Fisher, right? I'd say it's probably 1912. I won't look it up. But so starting in about 1912, Sewell Wright realized you could estimate the number of loci contributing to a trait, assuming Mendelian inheritance, and by a breeding experiment where you have two populations that differ, create an F1 where their mean is sort of halfway in between, and then create an F2 that's sort of the F1 inbred. That if Mendelian loci were causing the differences between these two populations, and those loci were fixed differences, Wright provided a formula to estimate the number of loci contributing to this trait as a function of the variance in the F2s and the means in the F1s. And it is called Wright-Castle estimation because Wright was a graduate student at the time, and Castle submitted a paper to Science that said, my graduate student says the following formulas are correct. And Wright is not an author on it. I don't think Wright is even mentioned by name other than Castle calling him my graduate student. And we have different models of scientific integrity in the early 20th century. But this was the first result that allowed anyone to estimate the number of loci involved in a quantitative difference between populations. It's really clever, it's really neat, and the answer it gives is always equal to the number of chromosomes in the species in the population. It always gives the number of chromosomes. That's what it gives. Actually, it gives something like the number of recombination events likely to happen in the organism. Sometimes it gives smaller numbers than that, but usually it just gives the number of chromosomes or the number of recombination events. And the reason is the experiment is really just a 2-generation cross. So if there are more than the number of recombination events, there are more loci than there are recombination events in the experiment, you can't separate their effects. A single locus looks like the effect of many loci that have not recombined between them. And therefore, what you're not estimating the number of loci, but you're estimating the number of, say, non-recombining blocks that contribute to your phenotype. And what you will discover is if you have anything like an interesting phenotype, every chromosome, every area of every chromosome seems to contribute. And that has been discovered over and over again. But had the number of loci been very small for the trait you were interested in, you could have found that number using Wright-Castle estimation in the 1920s. Questions about this?
I have a question. How could this be compatible with polyploidy in plants and like such weird like things, and like how like polyploidy has like non-additive or like additive dynamics on plant population genetics?
Yeah. So the answer is there's no such thing as polyploidy. Come on. This theory is applicable to a diploid organism that has recombination everywhere in the genome and in a regular, predictable way. And if you start adding in non-diploidness, particularly in a way that creates complicated patterns of recombination and may involve not even consistent numbers of ploidy levels, maybe not even all chromosomes having the same ploidy, because maybe some of them really have to go to 3 before they're viable, and whereas others stay at 4, others end up at 8. Once you start introducing that stuff, we can start writing down what we would expect under a specific model of ploidy dominance and their effects on recombination. But do not even try to apply this diploid simple theory. It won't help you. Make sense?
Yep. Yes. Thank you.
So, Mega, what do we got next? What do we want to talk about? So there's a weird something on 341. I want to see what there. So this is talking about the Shrimpton and Robertson analysis of the 3rd chromosome contributing to a bristle number. And it says in this experiment, they found 17 distinct regions, each contributing at least 0.3 standard deviations to the line difference. Their data suggest a highly leptokurtic distribution of effects with relatively few. What does that sentence mean? Because we're going to see these words a few times. A highly leptokurtic distribution of effects with relatively few regions producing most of the. What is meant by this?
So I'm not sure what leptokurtic means, but I would assume that it's just not an even distribution. So certain areas are like farther distributed versus others.
Yeah. So the term leptokurtic refers to the 4th moment of a distribution. You remember the mean, first moment, sort of variance, second central moment. The leptokurtic refers to the 4th moment. And a leptokurtic distribution is one where the center is very narrow and pointy, and the tails are very long. So when distributions are considered highly leptokurtic, it means the number of observations around the mean, there may be some, and there may be a lot, but there isn't a lot of scatter around the mean. And that the scatter starts happening slowly and slowly and slowly but you get very fat tails. So one of the first really careful experiments to estimate sort of the number of loci and the effect sizes of those loci in bristle number suggested that you don't actually have a Gaussian distribution of effect sizes, but what you have is a leptokurtic distribution, where there are a lot of things that have like zero effect, but for things that have non-zero effect, there tend to be way more outliers than you would expect from a Gaussian. So that you have a few alleles of very large effect, much larger than you would have guessed from the total variance, and a lot of things sort of stuck at zero. That's what a leptokurtic distribution looks like. A big chunk at zero, and then these ugly sort of tails moving away. And we are going to see this, well, maybe we won't, maybe we'll see how far we get into this paper. But that observation of the distribution of genetic effect sizes might be leptokurtic, meaning lots of things around zero and way more things of large effect than you would think from a proper Gaussian distribution, will be a feature of many of these sorts of experiments. Okay, where do I want to go next? We got to get this real quick. So Julie, the paradox of per character versus per locus mutation rates. What is going on in this paragraph that starts on page 341 with Lynch reviews estimates of, and we'll talk about what that symbol is, and then we'll go down to sort of 10 to the minus 6, 10 to the minus 5 stuff. So did you figure out what VM meant in this context?
It's the variant of.
Yeah. It is, we're going to call VM the mutational variance. And what it is, it's the additive variance. It is the sum of the additive variances that were introduced in the population this generation. So in general, you know how to calculate the additive variance of a locus. It's P alpha, P alpha 1 squared plus Q alpha 2 squared times 2. That's the additive variance of a locus. Now let's imagine for the moment, one of the alleles was introduced this generation. Call it the A2 allele. So that the additive variance of this locus is P, a number very, very, very close to 1, times alpha 1 squared plus Q, 1 over 2n, times alpha 2 squared, times 2, summed over all the loci that mutated this generation. That is the mutational variance, or the additive mutational variance. And because P is so close to 1, we're often going to ignore the contribution of alpha 1 squared and think of it just as Q alpha 2 squared, which is really just alpha 2 squared over 2n. So for much of what comes, you'll see Turelli and Barton writing alpha. And what they really mean is alpha 2 squared over 2n. They mean Q times alpha 2 squared from when we were doing it over 2n, because Q is 1 over 2n. When we're talking about additive, sort of VM type stuff. And we're going to talk about the number, and what's going to matter is the mutation rate over all the loci that contribute to this trait. So this is where some weird stuff happens. So they're going to talk about 2 experiments. One of them that is going to estimate the number of new mutations affecting quantitative characters affecting a phenotype and the number of new mutations at a particular locus. And what Turelli and Barton write is, if I pick my favorite trait, bristle number, height, weight, fur size, any kind of quantitative trait, and estimate the mutation rate to new mutations affecting that trait, on average, over lots of experiments in lots of different phenotypes, I find that the mutation rate to new alleles affecting that trait tend to be on the order of 1 in 100. That 1 in 100 meioses may give rise to mutations that affect my favorite trait. If I then, alternatively, if I pick my favorite gene, I don't know what gene my favorite gene is, alcohol dehydrogenase. If I pick my favorite gene and I estimate the mutation rate to things that affect that gene, either the gene's expression, how much of it, the gene's activity, if I just in general estimate the mutation rate to something I can observe about the gene, my estimate of the per gene mutation rate is about 10 to the minus fifth to 10 to the minus sixth for like every gene on Earth. So the mutation rate to a phenotype is on average 1 in 100 meioses seems to give rise to a mutation affecting my favorite phenotype. 1 in 100,000 to 1 in a million meioses give rise to mutations that affect my favorite gene. Turelli and Barton think that's a paradox. But can you see any obvious solution to it? You have a guess, Julie?
It's just many genes are involved.
Yep. If 1,000 genes or 10,000 genes are involved, this isn't a paradox. This isn't even close to a paradox if 10,000 genes are involved. That's what you expect. 10,000 genes are involved. 10 to the minus fifth mutation rate, 10 to the minus sixth mutation rate will give rise to a 10 to the minus second per character rate. Nothing surprising here at all. Like this. The issue is, does anybody know how many genes there are in a genome, in a human genome, in a fly genome? Any of your favorite organisms? The answer is 20,000. Flies, it's 10,000. There are only 10,000 genes. So, in order for this not to be a paradox, you have to believe whatever character you are looking at, mutations in almost any gene can affect it. Otherwise, you've got a problem. There aren't enough genes to go around for hundreds of characters to have a 10 to the minus second per character mutation rate unless most genes affect most characters. To a first approximation, if there are 10,000 genes or 20,000 genes involved in every trait is affected by every gene in the genome. There is no such thing as an autism gene per se. All genes are autism genes. They just differ in effect sizes. Some of them are bigger, some of them are smaller, but everything affects your trait. All of them do. If that isn't true, well, this is a great paradox, and you have to start thinking hard about weird experimental artifacts that would create this. But the simple interpretation is that most genes affect most loci. We'll come back to this again. Which also, of course, means, everything is infinitely pleiotropic. If you have a mutation that affects this gene, it affects hundreds of different characters. It doesn't affect a thing, it affects loads and loads and loads of things to some extent or another. All right. Maybe because we are just about out of time, we will jump to the same idea again in a little more detail. So maybe I'll just talk through it and jump to basically models of quantitative traits. So where am I? I want to be at like 352-ish. Yeah. What maintains heritable variation for morphological traits? I'm just going to talk through this because we've got 10 minutes left. And so if there is a standard model for a quantitative trait, it is due to Landy. And it was his PhD thesis. And the Landy model is fundamentally an infinitesimal model. There are many, many, many loci affecting every trait, that each locus contributes a very small amount of the additive variance, that there is selection that is stabilizing on the trait, that natural selection is acting to keep that people who have the population mean of the trait have the highest fitness, and people who are substantially away from the population mean have lower fitness. That's the sort of standard stabilizing selection model. And therefore, genetic variation is under mutation selection balance. That if you have a variant that's very far from the mean, natural selection will tend to eliminate it to push the individual's phenotype back towards the mean. And new mutation every generation is creating new additive variants. So that at equilibrium, heritability is determined by the relationship between the width of the selection function, Vs, the total amount, the variance, the total amount of new mutational variance, Vm, and the total number of loci that can contribute to the trait. And Russ showed as part of his PhD that you can get perfectly reasonable values of heritability with n's in the hundreds and VMs at values consistent with what we can estimate in nature and some reasonable assumptions about VS. And that will more or less go down the road. Kind of. We'll talk about Turelli's criticisms of it in a second. But the standard infinitesimal model is that any locus you're looking at is under mutation selection balance. Selection is acting to stabilize the trait values. The strength of selection is measured by the width of the stabilizing selection, and heritability ends up being a function of the number of loci and the mutational variance. It turns out you can get a couple different equilibrium out of this system. And one of the equilibrium involves when new mutations have a variance, alpha squared, that is, say, much smaller than the selective width. When that happens, you'll get a whole lot of like intermediate frequency variance contributing to the trait. Another sort of equilibrium out of this system is when the average effect of new mutations is highly leptokurtic, meaning there are a lot of variants that have no effect or very little, and a lot of variants that have very large effects on the trait. So these are sort of highly deleterious alleles. That a deeply leptokurtic distribution of effects will also result in very reasonable stable heritabilities, where the total genetic variance looks like the number of loci times the mutation rate times the width of the selection function. Here we're assuming new mutations tend to be large relative to the selection function width rather than small. And this is what the infinitesimal model like there's sort of two kind of stable results out of the infinitesimal model. You can get very common alleles if the distribution of effect sizes is small relative to the selection width, or you get these solutions where most of the alleles are very rare. They have very large effects relative to selection, so they're very deleterious. And Turelli argues, looking at a lot of different experiments, that if you look at the ratio of new mutational variants to total genetic variants, that most traits that you look at really are only consistent with the second sort of Landy equilibrium, that the genetic variance is being contributed to by a leptokurtic function where most mutations don't do anything, but some of them have large effects relative to selection and are highly deleterious and therefore very, very rare. He thinks that's the only sort of Landy model that is consistent with the data, but that model implies all traits have at least 1,000 genes involved or more. That Turelli rejects this model because he does not believe most genes affect most traits. He thinks it's sort of a silly idea that everything affects everything. The infinitesimal model really cannot explain what we know about heritability and what we know about standing genetic variation unless most traits have thousands of genes involved in them and most new mutations either do nothing or are highly deleterious and therefore very, very rare. And that model will give rise to very little high frequency variation contributing a significant amount of the variance. He will go on and favor other models of natural selection with, say, fluctuating selection components that will give rise to very high frequency alleles that explain large fractions of the variance. But if we accept, so if we are to believe in the infinitesimal model that that it is mutation selection balance, for most traits, the totality of the data is only consistent with a leptokurtic set of new mutational of new mutational effects and a large number of loci involved, meaning individual alleles of large effect will be very rare if they exist at all. These sort of results were known in 1985, 1988. What was not known in 1985, 1988 was 20 years of GWAS studies where no one can find a high frequency allele of large effect. They don't exist. in anything, just about. So if we fast forward to 2020, and we have in our back pocket that none of your friends got 15 Nature papers by finding huge alleles of large effect common for their favorite disorder, that they all got negative results over and over again, or maybe they got these tiny little sort of effect sizes. We would say all of that is completely consistent with Landy's infinitesimal model with a leptokurtic distribution of effect sizes and rare alleles contributing a disproportionate amount of the new mutational variants, and them all being mostly deleterious. And that if there are high frequency things running around, they have effect sizes much smaller than the selective coefficient. So they are really sort of very weak effects. So high frequency things must have very, very weak effects and or a whole lot of the variance is rare alleles. That's what the theory ought to convince us of. And nothing about the last 20 years is inconsistent with that theory. And there we have it. Anybody want to ask a question in the last 2 minutes?
I guess I'm curious how non-additive effects play into all this, because I guess one of the assumptions of the infinitesimal model is that all the effects are additive, but I think we know that that's not really the case. Is one way of sort of addressing some of the downsides of the infinitesimal model is kind of relaxing that assumption and saying, well, maybe there's non-additive effects.
So dominance effects, I can go after straightforwardly. I've got pedigrees. I know I can figure out who are full sibs, who are parent offspring. I know for a fact VD only affects full sibs, not parent offspring. So I can actually estimate the variance component of genetic of this, the dominance component of the variance very, very easily. And people have done that, and there are very few traits where there's a significant dominance component. It mostly most of the variance does not come out as dominance. Now, that said, what the flying fuck was, okay, so the dominance component we know probably isn't very large for most traits. But there is, of course, these interaction components. And in particular, there is an interaction component that I might be tempted to call the additive by additive interaction component. And additive by additive interaction components will appear to be strictly additive when the two sites are in linkage disequilibrium with each other. So the additive component of the variance, when I do family studies, may have over time been systemically overestimated by a conflagration of the additive with the additive by additive compartment. If there had been enough time for linkage disequilibrium to decay between all alleles, you'd be able to separate those out. But in general, you will often overestimate the additive effect of an allele by the amount contributed not only by that allele, but by things in LD with it, as you saw in some contexts, and by the interaction component between them too. So epistasis, additive by additive interaction, could very well have been misleading us for a long time about the fraction of variance that is additive versus the fraction of variance that is non-additive when they are near each other. More higher order epistasis tends to go away pretty quick with recombination. But if the sites are close enough that there isn't significant recombination between them, deconvolving the additive components from the interaction components is a challenge to something like impossible, given our current state of knowledge. Other questions? I guess we're over time already. So this is all fun. Test is on Thursday. You'll send me a cheat sheet. I'll send you the exam. You'll send me back the exam. When you send me back the exam, I'm going to assume you've closed out the class and that's it for you. If you don't send me back the exam in a timely manner, I'm going to assume you got busy and give you an incomplete, and we'll figure out when and how we'll get that fixed over the next few weeks or months. And if you are, God forbid, an undergraduate needing to graduate right now, turn the exam in pretty quick so that we make sure that we don't mess up any of your transcript stuff.
I have a question about a timely manner. Is that before you wake up on Friday?
No. A timely manner is whenever you can do it. I think I have to hand in grades sometime like in May 10th or something. So I think, but I might be wrong. At some point, someone is going to poke me and say, turn in your grades, Cutler. And at that point, I'm going to have to turn them in. And if I haven't gotten your exam back by then, I'll give you an incomplete. But I don't want you spending more than 4 or 5 hours on this. I don't want you spending more than 2 hours on the exam. But 4 or 5 hours is I think not crazy. But I know many of you are literally in the middle of your qualifying exam period right now. And I don't want you to fail your qualifying exam because you spent too long on my exam. So if it gets pushed off, it gets pushed off, and we'll deal with your incomplete after all this other nonsense is done. But if it doesn't, just do it soon, do it Thursday, do it Friday, do it Saturday, do it Sunday, do it Monday. I don't care when you do it. Sit down, do it for 3 hours, 4 hours, and then send it to me and watch that you got something like a strong A to maybe just a B of some sort. It'll be fine. But if for whatever reason you can't send me anything because you have no 3-hour block to sit uninterrupted and thinking hard, because that might happen, we'll deal with it whenever you do have time. I don't want you to fail your qualifying exam over this. But if you got a good block of 3 hours, just take the exam. Other questions? And don't kill yourself over this. Like really don't kill yourself. It'll be fine. You're doing great. Some of the problems will probably trick you. Don't worry about it. You got any last minute questions? Email me. Maybe I'll respond before Thursday. Maybe I'll respond after Thursday. Who knows? Talk to you guys by email.
Thanks, Dr. Cutler.
Thanks for everything.
Yeah.
Bye.
Thank you.
Thank you so much.
Thanks.
Bye,